All right, good morning. Thanks to the organizers for the opportunity to present some of our preliminary work as we dive into the era of third generation sequencing and a really long collaborative effort to explore the implications of prostate cancer risk genomics on regulatory function and the transcriptome. And here we're going to be focusing specifically on epigenetic modifications as a potential mediator of these transcriptomic changes. So we know that single nucleotide polymorphisms, or SNPs, can impact local methylation rates, and these are referred to as methylation QTLs or mQTLs. And there are a variety of different assays in which we can capture epigenetic changes at these CpG sites, such as array-based approaches. But what's really exciting with Oxford nanopore technology, or just nanopore sequencing in general, is that the base callers for these long sequencing platforms not only give you the DNA nucleotide information, but also allow you to simultaneously call the epigenetic modifications of those bases. So the 5MC and 5HMC modifications of those C cytosines and the CpG dinucleotides. And so this is really exciting for us, as we are really interested in looking at how prostate cancer risk genetics influences epigenetic changes in the prostate cancer tissue, and how this can help us interpret some of the genetic risk consequences at a functional level. So this talk has sort of two main components. One is going to be more on the bioinformatics side, and the other on the statistical side. The bioinformatics is really in trying to address some of the challenges in working with long-range phasing of this nanopore sequencing data, particularly when we have shorter reads than we are otherwise expecting from the longer read protocols. And then the second part is going to be going through some of the statistical approaches that we've been looking at in order to do allele-specific methylation QTL analysis in order to empower discovery of these SNP associations with epigenetic modifications. So just a very brief background on what we're really trying to do when we're doing mQTL discovery. So we have a polymorphism on the left-hand side that we're assuming is biallelic, so it has two different allele states, A and B. And we are assuming that it has some cis-acting effect on a nearby CpG site that is leading to epigenetic modification. So it's either going to be methylated or unmethylated. And so the idea is that can we associate the allelic status of these SNPs with the methylation status at the CpG site of interest? And if we are operating under the presumption of a cis-acting modification, then that means that the DNA molecule itself, or the haplotype, is particularly informative in the relationship between the SNP allele and the methylation status. So if we have haplotype resolution information, then we can decompose an individual into its two primary haplotypes and then look at the corresponding methylation rates of those specific haplotypes. Absent phase information, say if we were looking at trans QTLs, then we would alternatively look at the genotype dosage at the methylation at the mQTL SNP and corresponding methylation proportion at the CpG site of interest. So as I mentioned previously, this is part of our ongoing efforts looking at prostate cancer risk. It's particularly highly heritable cancer with heritability estimates of around 60% from twin studies. It's also highly polygenic with over 150 sites that have been identified from genome-wide association studies associated with prostate cancer risk. So we have this pilot data of 28 benign prostate tissue samples generated from Prometheon sequencing protocol with base calling performed with Dorado. We have variant calling using this Pepper margin deep variant platform. As I mentioned previously, when we have shorter read nanopore throughput, like in this context here, we have 5 to 10 kb. This provides a lot of complications in doing long-range phasing. We also have relatively modest sequencing coverage in our data, 15 to 20x. And so when we use standard phasing approaches that are read-based, such as long phase, we actually end up with much more shorter phase sets than we would otherwise expect from these phasing algorithms. And so the figure on the right illustrates the distributions of the phase set lengths for our 28 samples, where we see that the median length is anywhere from 10 to 25, maybe 30 kb. And given that a lot of mQTLs can be quite distal from the CPG site of interest, we want to dramatically improve this in order to be able to perform mQTL discovery. So we proposed a two-stage long-range phasing approach where we combine the readback phasing with statistical phasing that we typically use in the context of, say, genome-wide imputation. This allows us to use population-level LD in order to bridge the gaps between disjoint phase sets based off of reference panels from data sets such as the 1,000 genomes. We developed just a computational pipeline that takes the phase set information that we get from long phase and then extracts out the panel reference SNPs from that data set in order to perform population-based phasing, and then post hoc reinserts any of the novel variants that otherwise wouldn't be in that reference panel based off of phase set membership. Then we take those phase VCFs and then we do haplotagging of the methylation data within the original BAM file in order to get methylation-based haplotypes. So the second part of this is the statistical component. Now that we have the chromosomally phased methylation and SNP genotype data, there's a variety of different methods that we could use in order to do the mQTL analysis. We could ignore phase altogether, which is obviously not of interest, but this would be your traditional mQTL analysis where you associate SNP dosage with the methylation proportion. This could be done with using the count-based data, using just a quasi-binomial GLM, where we just have one observation per sample. We can alternatively perform an allele-specific mQTL analysis, where we would need to account for the multiple observations per subject, such as using generalized estimating equations or mixed models. And so for all the heterozygous individuals, we would have two haplotypes of interest and model those data accordingly. However, that's inefficient because we're only using the heterozygous individuals as part of that analysis. So we propose this alternative joint model that kind of encompasses both of these two separate approaches in order to not only incorporate all samples, but also unphased and phased data, because there are instances where if the CPGs of interest are in homopolymer regions, then we have a non-trivial number of unphased reads that are still informative of methylation status. So I'll just kind of briefly, I like figures for illustrating notation as opposed to just kind of slides of equations. So this kind of gives you a general understanding of how the data is structured. So we have our haplotypes of interest for people who have phase information available. We also have our unphased reads in terms of these kind of duples of the methylated number of reads and the total number of reads that we're kind of framing in this binomial kind of representation. And then we want to model all of this data simultaneously. So the way we can do that is just envision that we have a binomial representation for all unphased reads, where we have the allele status as the kind of exposure of interest, and this would be just through kind of a logit based link function. And then we also have all of the haplotype specific data as well. We just have to account for the, in this joint model, the representation of dosage here. So when we are modeling unphased data, we are using the actual genotype dosage. So we refer to that as kind of a 0.5 and 1 values versus when we're modeling allele specific, we just model that dosage as 0 or 1. So that reflects the proportion of the alternate allele in the regression framework. So we ran some simulations. I won't go through all of the different settings and details. Suffice to say is that With the joint-based approach, and we generally have really good control of type 1 error. There's some minor inflation when we have smaller sample sizes, or when the base methylation rate is closer to 50% versus more towards hyper or hypomethylation. But what we see on the power side of things is that we have dramatic increases in power when we use this joint approach because we're using all available data, as opposed to just focusing on allele-specific methylation analysis or using just the kind of the total methylation using this phase agnostic strategy. So I mentioned our pilot data, and so even though our sample size is fairly small, we wanted to kind of explore the possibilities of replicating known mQTLs in prostate tissue. So we turned to Dye et al. Was a publication from 2020 that was using methylation array data to explore mQTL associations at 147 known prostate cancer risk loci. And one in particular of interest that was not only was it mQTL, but demonstrated associations with transcriptomic dysregulation was at IRX4. It's convenient for us because there's a very high minor allele frequency in terms of the corresponding risk SNP. And in our data, since we have both HMC and MC epigenetic modifications, we could do this association with both. And we see a nice peak at the locus of interest, which is a DNase hypersensitivity site. So very encouraging to see that this works out very well with our joint modeling strategy. However, there are also some limitations. Not everything is a home run. And this is in part, we see here in this figure, it's another locus at TBX5. The variant of interest still has a relatively high minor allele frequency, but we're seeing kind of poor performance here in terms of likely type 1 error inflation. There's a lot of the p-values in this kind of Hudson-style Manhattan plot are kind of running away from us. And we see that this is often due to situations when we have hyper or hypomethylation, which is leading to separability issues in the model fit. And so this is causing not only biased mQTL estimates themselves, but the downwardly biased standard errors from the small sample setting. We can kind of clean this up and through catching the warnings and errors and defaulting to the phase agnostic methods when these situations arise. But it's not really all that satisfactory and from a theoretical perspective. So we're looking at different methods that would either be Bayesian or mixed model in nature in order to try and stabilize some of these estimates. The literature has shown that ultra-long nanopore reads can lead to very large phase blocks. But when we're dealing with samples that have lower DNA integrity and our average read lengths are more modest on the nanopore side of things, then we find that this two-phase hybrid strategy of both population-based and readback phasing can dramatically improve the phasing that we see in our sequencing data. We've outlined this statistical framework for kind of leveraging all available information, both phase agnostic and phased information for allele-specific mQTL analysis. But we see that the GE-based approach has some issues, and we're kind of exploring other statistical methods in that framework. And then finally, this is all sort of prelude to kind of more interesting work that we're excited to pursue using paired DNA, RNA, nanopore sequencing, where we want to do allele-specific methylation expression QTL analysis. So being able to jointly phase all of the DNA and RNA information and look at how methylation is mediating transcriptomic dysregulation at an isoform level. So with that, I would just like to thank my colleagues both at Mayo Clinic as well as Moffitt Cancer Center, and acknowledge some of our funding that supported this work.
Oh, cool. So the variants that overlapping with DHS, since you have base pair resolution, can you do the footprint and find exactly what TF binding sites were disrupted?
Yes. Yeah. So yeah, we have all the positional information. So,
Yes, exactly. Yeah.
Yep.
Okay, cool. So the other thing is I was just curious for any known prostate cancer related genes, the difference between mQTL and the QTL, how many like percentage of the sites you have differential methylated sites in addition to the QTL? You probably have shown it.
Like the versus the EQTL?
Yes, yeah.
Yeah, that's a good question. I mean, so the original, that Dye paper I cited, they saw that most mQTLs were EQTLs as well, but not the other way around, because obviously different.
I just wonder the difference percentage.
Yeah.
All right, cool, thanks.
Great talk, thank you. So on the joint modeling, there are methods that have been used for EQTL and histone, like chromatin accessibility QTLs and so on and so forth, such as Rascal and all. So do you think those are not applicable to here? That
Are you talking like the like the
genotype
expression?
and allele combined together?
Yeah. So the trecase, I think, Wei-Sun, I think, has.
There are a couple of methods, like Rascal is one, I think, for chromatin QTLs and for ataxia QTLs. People have done this type of joint modeling. Of course, not with long reads, you have more power here in terms of phasing things, but I don't know if they are applicable in your opinion.
Yeah, so I know that there, at least with the expression ones, there are fundamental differences that we had to sort out, because that was some inspiration that that we took from in terms of doing this analysis, because of it's all DNA based, right? But I think there are, we have come across other allele-specific approaches that we're looking at. And so I know I've seen some mixed model approaches that have been developed. Our hope was that the computational efficiency of GEE would be really helpful here, and its robustness with the sandwich estimator. But then when we run into the small sample issues, there's like not much recourse. So it's either invent a Firth-corrected binomial approach or see what else we can do, or just stomach the computational limitations of Bayesian or mixed model issues.
So one other quick question would be, if you looked at the genotype dependent QTLs and then looked for those that have enough reads that you can statistically do something, and then went and looked for the allele specific patterns, so roughly what fraction of those you would think that there would be allele specific signature or like trend in the same direction?
Among like just heterozygous samples, or?
Yeah, among the heterozygous samples.
Yeah.
I mean, I know your sample size is small. We are dealing with the similar things ourselves.
Well, at least with our data, I know that the amount of unphased reads we were seeing, even among heterozygous samples, like on a per CPG basis, was anywhere like as high as 30%. So there was definitely, among those that were heterozygous, there was still 70% of those reads on average were, phase informative in terms of the, being allele specific with methylation. But we were still tossing out all of that unphased information. So the analyses that were reflective of the ASM models were using just those phased reads, but that's.
All right, cool, thank you.
Thanks.